Today's topic is about specificity of enzymes. Enzymes are basically biocatalysts, but these enzymes are showing particular specificity and they are also classified depending on their activity. So one that is absolute specificity means that enzyme is absolute specific for that reaction only or absolute specific for the substrate only. Like example, hydrolysis of urea to ammonia and carbon dioxide is catalyzed by that urease enzyme, but this urease enzyme is absolute specific for the urea only. If urea is present, then only this reaction will occur, otherwise this reaction will not occur. So thiourea, though structurally similar to the urea molecule, will not act as the substrate for the urease enzyme, and that is showing the absolute specificity of this enzyme urease. Similarly, glucose oxidase will oxidize only beta D glucose and no other isomeric form. So this enzyme show absolute specificity, and that is giving advantage for the some particular work. Like glucose estimation in clinical biochemistry laboratory is done by this glucose oxidase, so it can accurately measure whatever the glucose which is present in our blood, that is particularly beta D glucose, and we can accurately measure our blood glucose level. Same way, bond specificity. Here, most of the proteolytic enzymes are showing bond specificity. Proteolytic enzymes towards the breaking down a bigger protein molecule which we have taken in the diet into a smaller fragments, and that is possible by these particular proteolytic enzymes. So here example, trypsin that can hydrolyze peptide bond formed by carboxyl group of arginine or lysine. Arginine or lysine, that is amino acid present in protein, then only this trypsin is able to break down that particular protein. Otherwise, this trypsin is not able to work over that protein. So this trypsin that is showing a bond specificity for the activity of the enzyme. Same way, group specificity means one enzyme can catalyze the same reaction on a group of structurally similar compound. A best example, that is hexokinase, that is one of the key enzyme of the glycolysis. So hexokinase that can work over the any hexose molecule, so can catalyze phosphorylation of glucose, galactose and mannose. So glucose phosphate, galactose phosphate and mannose phosphate can be generated. So that is a common activity over a group of molecule. So it is enzyme showing a particular group specificity. So absolute specificity, bond specificity, group specificity. One more, that is stereo specificity. Human enzymes means what are the enzymes present in our body are specific for the L amino acid and D carbohydrate, means our enzyme is able to act over these L amino acid and D carbohydrates only. If D amino acid and L carbohydrate is present, then our enzymes are not able to work. Example like fumarase, that is enzyme will hydrate fumaric acid to the malic acid, but the corresponding cis form, means cis form of the fumaric acid is present, then this fumarase is not able to make malic acid. So malic acid will not generate it if there is a cis form of the fumaric acid is present. Same way, lactate dehydrogenase convert pyruvate to the L-lactate, but this enzyme is not able to make D-lactate molecule. Same way, as a specificity, you have to remember about the chirality of the enzyme. So these enzymes are also able to identify specific chiral center. Suppose that enzyme is acting over the third carbon of molecule, then it will act over the third carbon only. The action or reaction will not take place over the first carbon. That is showing a chirality of the enzyme, and this is very important for the understanding of the various drug activity also. So some drug like ibuprofen is active in S plus form only. If any other form, then this enzyme is not, this drug is not able to work properly. Some other chiral form of drug is sometimes dangerous, like S isoform of the thalidomide, and that is also one kind of specificity of the molecule in relation to the enzyme activity. So that you have to remember: absolute specificity, bond specificity, group specificity, stereo specificity, and you have to remember about the chirality. So that's all about the specificity of the enzymes.